Hello and welcome to the Discovering and Developing Drugs revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. Traditionally, drugs were extracted from plants and microorganisms. There are many examples of this, such as penicillin, which was discovered by Alexander Fleming. Foxglove plants contain a drug called digitalis. This is used to treat heart failure. The bark of a willow tree contains a derivative similar to aspirin, which is used as a painkiller. And penicillium mold produces the drug penicillin, which is an antibiotic that treats bacterial infections. The starting point is often a chemical extracted from a plant. Once the active ingredient within the plant has been identified, then most new drugs are synthesized by chemists in the pharmaceutical industry. Why new drugs are tested? New drugs are extensively tested for toxicity, efficacy, and dose. To check the drug is safe and not toxic, to check its efficacy to see if it's effective, and to find the optimal dose or the lowest dose that will be effective. Peer review. Before the results of drug testing and clinical trials are released to doctors, scientists or the public, they must be checked by other independent scientists to make sure the research is valid, reliable, accurate, not biased, and reported honestly. This checking process is called peer review. Only after this scrutiny can the results be published in a scientific journal. Drug testing. There are two main stages to testing drugs. The first stage is preclinical testing, and the second stage is clinical trials. Preclinical testing is done in a laboratory using cells, tissues, and live animals. For cells and tissues, the drugs are first tested on cells or isolated tissues in a lab, used to check toxicity and whether it seems to have the desired effect. Those that pass progress to the next stage, any that fail are discarded. For live animals, the small number of drugs that passed the previous stage are now tested on animals to check safety and how the drug behaves in a whole organism. Clinical trials is when the drug is trialled in humans. Only drugs that pass all of the previous stages will be tested on humans. There are two main stages to clinical trials. Firstly, we use healthy volunteers, and very low doses of drugs are given to healthy volunteers. This is mainly to check for side effects and basic safety. Next, it will be tested on patients, assuming it passes the healthy volunteers stage. Once the drug has passed all of the previous stages, the drug is tested on a group of patients. The dose is gradually increased to test effectiveness and to find the optimum dose. Double-blind trials. The people in the trial are split into two groups. One group will receive the trial drug, whilst the other group will receive a placebo, which is a dummy drug. Neither the patient nor the doctor knows who has what. Double-blind. This is designed to prevent bias. Information about who received the real drug and who received the placebo is only revealed after the trial has finished. Thank you for watching. See our revision notes, the link is in the description.